Good afternoon, everyone. This is Wafik El-Deiry. I'm the director of the Legorreta Cancer Center at Brown University, coming to you today with a Legorreta Cancer Center Directors podcast with my colleague, Dr. Robert Sobol, who is joining us today. Rob Sobol is our associate director for basic science at the Legorreta Cancer Center and co-leader of our cancer biology program. Welcome, Rob.
Thank you for having me today.
It's a nice fall day here in Providence, a little bit rainy, a little bit on the cold side. But as we walk around the jewelry district, we see some exciting things going on with the Danoff Life Sciences building going up, where you can literally see progress every week. Um, at our cancer center, some exciting things are happening as we have been going through our strategic planning and making progress there. And as you look around the jewelry district, there's other excitement with the building going across the way there, the state building
The
with
public health building, yeah.
and with the Ocean State Labs. So, uh, you know, let's start with you telling us a little bit about yourself, your interest in science, some of your experiences, and maybe some of the things that shaped your career path.
Happy to. And again, thanks for letting me join today. My story starts back in Long Island, New York, where I grew up. And like a lot of, I guess, firstborn children, the idea was, oh, you should go to medical school. So I was always very interested in the sciences all throughout high school. And at Allegheny College, that was my original plan, is to go to medical school. But one of the probably the most prominent thing or the most exciting aspects of undergraduate college was the biology and chemistry labs. And so instead of going down the path of medical school, I ended up in graduate school, also in Pennsylvania, but this time in Philadelphia at Temple University, where I received a master's in chemistry and then a PhD in biochemistry up at the medical school. And although at that time there were MD, PhD programs just starting, and so when I finished my MD, I finished my PhD, they invited me to participate in the MD program, but I thought, oh, 4 more years, I don't know if I want to do that. So I continued postdoctoral training. All during my graduate work, I worked very closely with Sam Wilson, who was an NCI researcher at the time. And I ended up working as a fellow with him, both in Texas and then at the NIEHS for quite some time as a research fellow. And it was at that time I started my own career, my own laboratory at the University of Pittsburgh Cancer Center, where I was for 12 years. Had the great opportunity to help shape a small cancer center in Alabama at the University of South Alabama, the Mitchell Cancer Center. And I worked there for some time helping to build that program, and then ultimately was thrilled to join here at Brown at Dr. El-Deiry's insistence 3 years ago. It was quite interesting doing all the interviewing during the COVID period, mostly Zoom, but I ultimately visited and signed, and then I've been here now 3 years. So it's been an amazing journey to get here and continue my work here and help build the Legorreta Cancer Center.
Thank you, Rob. One of the things that we as faculty really enjoy doing is our involvement with trainees.
Right.
And maybe you could tell us how that has unfolded in different parts of your career.
Thank you. Yes. Well, and in University of Pittsburgh, I was a brand new assistant professor trying to train students while I was trying to figure out what are we going to be doing in the lab was a bit of a challenge, but we did ultimately have consistently undergraduates come for as part of a Howard Hughes program where they would train them in molecular biology techniques for a semester. That was quite fun, but it was somewhat limited, although we did have postdoctoral fellows and others who trained in the lab. And some of them have gone on now to their own professor appointments or industrial appointments, depending on the individuals. Alabama, we had very few students, but there were a number of students who rotated through the labs, and I had several postdoctoral fellows and grad students. One of the grad students actually came with me here and ultimately finished her PhD here at Brown, and she was very excited about that. And she's gone on to bigger and better things now. And here at Brown, it's been quite an interesting path because the undergraduates are so hungry for research experience. I get about 20 requests a semester. Can I work in your lab? And sadly, I can't take them all, but I've taken about maybe 15 or maybe even 20 since I've been here. And I have to say, these are the smartest kids and so dedicated. One of the holdovers from COVID, but more so because I feel responsible, as I ask all the students to just text me when you show up and text me when you leave. I'm not counting hours, but just to know that there's someone in the lab. I get texts from various students, even this weekend, Saturday mornings, Friday nights, Sunday evenings, oh, I'm going to come and study while I'm running my gel or other, experimental procedure that takes hours. And I'm really enjoying that here. I've had now 4 or 5 honors thesis students through the past several years, and it's been a great experience.
Yeah, I like that idea, Rob. I usually tell the students, I don't keep track of when you
No.
clock in or clock out, but I know how long things take. Um, so, you have come up upon 3 years in Rhode Island, and as you know, we have high cancer rates here in Rhode Island. And you have really unique expertise to study that and to have impact in different ways. But what are your thoughts about, or how are you planning to address the burden of cancer here in Rhode Island?
Great question. It was shocking to learn after arriving the abnormally high incidence of both brain cancer and bladder cancer, among others, here in Rhode Island, ranking us in the top in the nation. That's not something you want to be the top in the nation of. And those are, in many cases, at least with bladder cancer, we would imagine it's related to environmental carcinogenesis. That is, there are contaminants in the air and the water and the soil that residents have been experiencing for decades, and that could be participating in the onset of those cancers. That's certainly one area that we're very much interested in. My background includes study on the genes and the gene products that are responsible for maintaining your genome. So we need an intact genome. Otherwise, if genes move around in the wrong way, we end up with various diseases, cancer being one of them. And so trying to understand the burden of cancer through that lens is a big aspect of what we're trying to do.
Tell me a little bit more about exciting research you're involved with, and maybe touch upon what are some of the exciting developments in your field.
Right. So, as I mentioned, we're very much interested in understanding those mechanisms that keep our genome intact. And as you can imagine, it's quite diverse. There's on or about 250 genes involved in that, and depending on how you count them, it could be as many as 600 genes that are constantly engaged in maintaining your genome. And as you might imagine, this is a very complex scenario. If you lay out in the sun too much, right, that's considered a predisposition to some type of cancer, or if you're exposed to radiation, or if you're exposed to a chemical. These are all various types of genome damage. And so we call them pathways. There's groups of genes that are responsible for these various types of exposures, and they actually all depend on each other. And we can take advantage of that in the clinic. And so people have done that. Many of you have probably heard about the drug that we refer to as PARP inhibitors. These are drugs that inhibit a particular set of genes involved in the DNA damage response. And they seem to be quite selective for things such as the BRCA mutant cancers that emerge in ovarian and breast cancer. And there's been some estimates that every single cancer has one or more defects in DNA genome maintenance pathways. And so can we exploit those defects to come up with very sensitive drugs to selectively target the cancer? Sort of the same way we do with bacterial infection. We selectively target the bacteria. It doesn't hurt the rest of the patient, with the exception in that case of a tummy ache. But in general, we want to try to target the cancer in a way that doesn't affect the normal healthy tissue. And that's some of the aspects that we're working on, in particular, associated with PARP inhibitors and related pathways.
So, Rob, you mentioned there are hundreds of these potential targets for cancer therapy, from DNA repair proteins to polymerases to helicases to all of these different targets. How do you decide what to focus on?
It's a great question. One of the tools that we've been building in the lab over the years, which we've now really focused on strongly here at Brown, thanks to some of the great technology advances that we have in our core facilities, is to identify the exact proteins, if you will, the gene products that are involved in the responses. And that's referred to as proteomics. And we've been building that technology in the lab so that we can selectively address those particular proteins that are involved in the response, uncovering for us new targets. And then in parallel, then we try to develop ways of screening drugs and small molecules that we can target those, selectively inhibit those targets.
How long does all of that take from the discovery to and do you see that this could be individualized to specific patients, or are you looking at a way to treat, a whole subset of patients?
Right. I think right now it's more of a subset. So as for example, with the BRCA mutant tumors, right, which are maybe 5% breast cancer and maybe an equal number of ovarian cancers, also expanding now into prostate cancer at some fraction. Those are pretty broad numbers. And so if we could develop targets that affect those mutant cancers, that's a pretty tall order. It's still considered personalized therapy in a sense, because we need to understand the tumor and how it's defective. But if we can expand into that more and more, I think that would be helpful. In terms of the time it takes, the more we advance our technologies, I think the more it'll go faster. It takes us about probably a year and a half to 2 years to identify in a really specific way the protein targets that we're interested in, but we're moving faster on that as we go forward.
Thank you. Maybe you can say a little bit more about how we learn more about causes of cancer in Rhode Island. And also, I know, over the last couple of years, you've been really doing a lot to organize interdisciplinary types of retreats and events. And, why is that important? And how does that help?
That's a great question. We try to do a lot in my own lab, but we can only do so much. Science has gotten so specialized. If we need to bring together bioengineers or structural biologists or RNA chemists, we're very fortunate that we have such expertise here at Brown. And not only do we have the expertise, but we have this enthusiasm to interact, because I think we all seem to realize that in especially in science, two and two makes five. And so you really need to work together to be able to answer questions that you would never be able to do on your own. So for example, we're built a team now that we're trying to develop a grant submission that involves computer biosystems analysis, engineering in terms of the computational analysis, chemistry, and then the biology that we bring to the table. None of the other four things would I be able to do in our own lab. And so having these other investigators here at Brown who are interested in partnering allows us to address questions that we otherwise would never have been able to attempt.
And specifically with regards to Rhode Island and some of the cancers you mentioned earlier, bladder cancer and brain cancer, can you just tell us a little bit about what is going on to learn more and to have impact here?
Right. So those are two areas that we have a lot of strength. Brain cancer, we have folks like Sean Lawler who bring such a great diverse perspective on the translational science associated with brain tumor biology. And then we have fantastic clinical folks, and a lot of places have great clinicians. And what you want is great clinicians who are interested in the science behind that. And probably every single clinician I've spoken with here at the Cancer Center is fully engaged in the research questions and want us to participate together and join together, such as Clark Chen and his clinical work on brain cancers. We have a whole team being built now around the fantastic clinical work on bladder cancer that has been going on for 4 or 5 years here at Brown. And we're trying to bring to the table then basic biologists, and analytical chemists so that we can analyze the samples in a more detailed way. Kurt Pinnell, for example, in his mass spectrometry analysis of small molecules, can give us an understanding of the contaminants that are potentially cancer causing here in the soil and in the air and in the water. So this only comes about because you have people that are excited to interact and work together.
Thank you. A cancer center reaches into the community, and this cancer center cares about the community in Rhode Island. And I'm wondering, what are some of the things you are doing or have been learning or would like to share with members of our community here in Rhode Island?
Well, one of the challenges for any academic scientist, professor, or institution is let's say the problem of being isolated from the community. And one of the several of the events that have gone on since I've been joined here at Brown and the Legorreta Cancer Center are these community interactive meetings where you get to talk to the community, and not to lecture the community, but in fact it has to be, we want to hear from them. And I participated on several of them, and it's really enlightening because that interaction has to be two-way. And so we need to understand their concerns and their fears, and sometimes things that are going on in the community that would have an impact on the cancer burden that we would never know by just keeping our nose in the academic ivory tower. And so participating with the community members like this is the best way to do that.
And what would you want our colleagues elsewhere in the state or at nearby institutions as far as our culture of collaboration and what's possible here?
Well, one of the things is, as I mentioned, not only are the students just fantastic in terms of their intellectual capacity, but also their dedication and their engagement with research. They're not just coming to work in the lab so they can get a letter and go on to another, grad school or med school. They really want to work. And then that message or that sort of enthusiasm permeates through all the colleagues and researchers and clinicians that we have across campus. And in the 3 years I've been here, there's not been a single person who has said, no, I don't want to participate in that. And whether it's a analytical chemist or a structural biologist or a systems biologist or a computer programmer, they all seem to want to try to work together for the bigger picture that the Cancer Center is trying to bring light to.
Can you say a little bit more? We have some great colleagues at University of Rhode Island, and can you tell us a little bit about some of the interactions that are going on there and what the future might look like?
Well, one example I see almost on a regular basis is my colleague and co-director of the cancer biology program, Pat Dubaleka. And talk about a dedicated person. She is so enthusiastic about her work. She seems to know everybody and everything that's going on here at Brown. Granted, she's been here, I think, 10 years. But she works hand in hand with some of the clinicians she studies mostly lymphoma. And that just brings such a strong, she brings such a strong, let's say, level of enthusiasm and collaboration that makes it such that I want to do the same. And the other group too, which we haven't mentioned yet, is the folks at URI, University of Rhode Island. They have such a powerful set of faculty that they've been recruiting over the years. And they're all very dedicated and interested in interacting. In fact, we have one of them coming to visit, I think, later this week. They participate in all of our cancer center meetings and our cancer biology, our cancer biology program seminars. And so we have regular conversations about their science and our science and how we can work together. And hopefully that'll lead to some grant submissions in the coming year.
Rob, we know that this is a difficult time in academia with funding for research in science. How are you doing at this time, and what would you say to young people who are thinking about careers in science?
Right. Scary question. It is probably the period in my career that's the biggest challenge because of the funding rates at the National Institutes of Health and the stress that puts on all the foundations that also provide funding, although it's obviously a much smaller level of funding. So what's healthy to see is we still have let's say young fellows, postdoctoral fellows, for example, many of which are deciding I want to go into clinical research or I want to go into corporate research. But you still have that fraction that's saying, we just interviewed a few weeks ago, several postdoctoral fellows several weeks ago, and one of them in particular, I'm interested in academic research. That's it. I don't want to think about anything else. And so there are those folks out there, and I think you have to be very dedicated because it's not an easy path, but it's a rewarding path. And I look forward to finding you and having you come and join us here at Brown.
Rob, what's the importance of work-life balance, and tell us something about yourself that you're interested in outside of work?
Well, that's a great question because up to a few years ago, I don't think I really had a great work-life balance, and I don't think that's healthy. So moving back here up north, kind of felt like coming home. As I mentioned, I grew up in Long Island, which is just across a ferry ride away. And I decided I really need to have a good work-life balance. And so I regularly enjoy, like tonight, we'll be playing pickleball. I do that often also with Howard Safran, our clinical collaborator here and head of the experimental therapeutics. Um, Skiing I took up again after 25 years. Now that I'm no longer in the South, there's a lot of skiing around the area, and golf over the summer. So those things keep me quite busy in the, when I'm not trying to write a paper like I sat at my desk till 8 o'clock last night before going home.
Sounds like fun. Is there anything else you'd like to say that we haven't touched upon that you think is important for our colleagues or for our community to hear?
Uh, I I just want to say, like you mentioned, this is a very difficult time in academic science. There's cutbacks everywhere across the government, and making it difficult to fund the work that is so essential for bringing cures to the community. I think one of the things that we tend to forget is, as a community, is that this is an essential part of the fabric of our community. So we need help, and we need to work together with our local governments and our community members to um, find the resources so that we can continue to build this cancer center through research dollars, through administrative support, and also to help the students and the fellows and the trainees that we need to invest in our future.
All right. Well, Rob, thank you so much for stopping by this afternoon.
It's
Been
been
a
really fun. Thank you.
fun interview, and look forward to seeing you later today at some important meeting we're supposed to be at.
That's right. Okay, thanks so much.
You're welcome.